Hi there. So today I'm so excited to be joined by Dr. Ron Davis, who is chair of the Open Medicine Foundation Scientific Advisory Board, as well as the director of the Stanford MECFS Collaborative Research Center. Today, I just wanted to talk a little bit more about your research and focuses in MECFS research. So thank you so much for joining me.
Oh, you're welcome. Very glad to do this.
Excellent. So I wanted to start off talking about some of your work, which focuses on genetics. So a lot of what you do focuses on using genetics to look if there are pathogens that may be involved in this disease. So I'm wondering if you can explain more about what this means and why this is important in MECFS research.
Well, the pathogens are really important. For one reason, we know that this disease is almost always caused by a pathogen infection. There are a few cases that seem that didn't involve an infection, but in fact, sometimes it's really difficult to be sure of that because there are some infections that go silent. I mean, people don't realize they've been infected. So the fact that it gets started by an infection is really important. However, that infectious agent is probably completely gone in most cases, not all. But the disease really looks like an infection in many respects. In other words, when you get an infection, you're fatigued. Mm-hmm. So it's really important to figure out whether that is a possibility of an infection. And the reason for really sorting that out is there's almost always a cure for most infections because people have worked on this for hundreds of years. Mm-hmm. So if we can find an infection, we can probably treat it and we can probably cure it. So that's why it's so important to do. And people have looked and haven't found anything, but we keep feeling that we should use the best technology that we can come up with to keep looking. Just as for example, there's a possibility of a trypanosome infection.
Right.
Now, there aren't any known trypanosomes in the US that infect people. That doesn't mean they don't exist. There are trypanosome infections of horses, and people have been known to get infections from their horse. It's rare, so probably not this is not causing it, but just as an example that is an organism that is an infectious agent, and it causes something called a sleeping sickness. Now, patients might be understanding they have sleeping sickness, right? I mean, and sleeping sickness in this case is defined really as the fact that they change their light-dark cycle for a trypanosome infection. That's why it's called sleeping sickness. And they stay up all night and sleep during the day. That's exactly what happens with MECFS patients in general.
Right. So
it's just a possibility. So we've developed DNA probes that we can look for it in people. It's a long shot. It's just something that has to be done to try to make sure that we can't find that. And if we can't find it, it doesn't mean it's not there. And that's always the situation. Because for one thing, where is the infectious agent? Basically, all we can do is saliva and stool and urine and blood. And we can look in those for this, but if it's somewhere else, we might not see it. Very likely, if it's any place that might get some dead organisms may get into the blood, and so we still might find it, even though it's not the primary site. That's just an example. And so we have made probes for all the DNA viruses, almost all of the RNA viruses, all the parasites, and we are looking to see whether we can make things for the funguses and bacteria. Those are a little trickier because there's an awful lot of them. But that's just a program that we have. It's a small program in the sense that we have one researcher that is really good at that sort of thing. That's part of his projects. He has other projects he works on. So it's a matter of getting patients in and having all these samples to look at. And it is a slow process because we don't have enough people and we don't have enough money. But it's a commitment that we've made to try to look through all of them that we can. So that's what that program's about. It looks for the presence of the nucleic acid. RNA is harder because it's not as stable and won't stay as long. And in order to do that, we have to convert the RNA to DNA, and the DNA then is tested.
Wow.
Now, how we got into this was when the pandemic hit, the labs were closed.
Mm-hmm.
So we had to stop doing research except for reading and writing papers and writing grants and that sort of things happened, but they weren't experimental things. Except this one individual, his name is Tadang Shen, decided that he could get into the lab and work if he worked on COVID-19. So he decided that he would work out a better assay for the virus as a PCR assay. And that was an RNA virus. So he said that would allow me to develop the technology for doing other RNA viruses, which we want to do to look in the patients for. So that allowed him to get into the lab, and he did. He worked out a really nice method for doing it. It took a little too long, and it was mostly very simple. And it was called a one-tube assay, as opposed to making multiple steps. You could do it all in one tube. So it made it simpler, and it's used at Stanford, but other people have developed their own ways of doing it, even though it's more complicated. They don't like to adopt another method. So we haven't pushed it that hard, but it's taught us a lot about how to do this. So that's the infectious idea.
That is so interesting. I think also what you said before was quite interesting, how sometimes it's just because you can't see an infection or a virus doesn't mean it's not there. And I think that reminds me a little bit sometimes. I know with Lyme disease, for example, there's a spirochete that causes this disease, and it could be a very difficult bacteria that hides. And I know that makes it even more difficult, not only to find, but I believe to treat, if that's correct. And it just goes to show that just because you don't see something immediately doesn't mean it's not there.
That's right. That's correct. We have probes for that organism as well, and others.
Oh, do you?
So we basically are going through the infectious disease list and trying to do everything eventually.
Right. I think it's also just so complex with ME/CFS, specifically these patients, because they can be triggered by so many different things. And it's not a one size fits all trigger. So one person might have Epstein-Barr virus, and the next person might have been, COVID now, or even sometimes you see Lyme disease or any. So it's even more difficult, I think, because there's so many different origin stories for this disease.
Or Ross River virus. I mean, there's just so many different ones.
Right.
That's right. But the other issue is that this disease appears to be almost always started with an infection.
Mm-hmm.
And so what happens in an infection is a different topic. But we want to see what's going on in the very early stages, if we can. And that may give us the clue as to what's happened that keeps this around. Most people, when you get some ailment, will go back to normal after a while. So either you haven't cured the infection or something else has happened that sustains the symptoms. Right. And we need to sort of try to figure out what that might be. Fortunately, with COVID-19, there's a lot of doctors are now taking that one serious, and NIH is taking it serious for the first time.
Right.
But I'm not wanting to work on it because I don't have any specific funds for COVID-19.
Yes.
All I have is ME/CFS funds from patients, patient families, or other people that want to support it. I'm not going to use that money for COVID. I have a small amount of money for COVID-19 work directly, which is to look to see what kind of viruses are reactivated.
Oh, that is interesting.
And it's been extremely difficult to get COVID-19 samples, surprisingly.
Really? Yeah.
And when that's because they are in emergency rooms or hospitals, and the physicians that see these patients sort of have first access.
Right.
And then I want to get some samples from them, and they say, well, no, I need them all.
So there it becomes more, you wouldn't think it to be difficult, but it is.
Finally, I now have some samples from Sweden, of all places.
Oh, wow.
From Bergquist, who's one of the other collaborative centers.
That's right, because I know they're looking into the ICU COVID patients over at Uppsala because they're collaborating, I believe, with the neurointensive care unit at Uppsala.
Yeah, that's right. So that's where our samples are coming from.
Okay, yeah. I think that's just that also like is just this unprecedented opportunity to look at this disease as it progresses in an early stage. So thanks for explaining a little bit more about that. Kind of on a similar vein, a lot of your work and interest has been in creating low-cost technology. So I'm wondering if you can share your ideas of what you would like to see in helping to diagnose ME/CFS and more about the technology.
Well, we would love to have a very sensitive test that has a low level of false positive, false negatives. I've been a big proponent that you need to take those seriously and try to figure out why you have them and eliminate them. Also, I know that cost is an important component. Now, the problem is I don't have total control over the cost. Mm-hmm. I can make the device very inexpensive and the reagents very inexpensive, but I can't control what the clinical lab will charge for it. So in other words, I could give them a device that costs them $1, mm-hmm. and they may charge 100 because they can.
Because they.
I don't want to put them down, but that is always a problem.
Right.
They may not do that because it is a little bit, I think, unethical. But all I have is the control over the original cost. So we do need a diagnostic, and that is one of the things that we have been working on for a while. But the device has to be very low cost. And so we have something called the nanoneedle that is very reliable in detecting it. Healthy people don't show this. All the patients do. Again, we have not been able to look at patients with other diseases because I'm not a doctor. And so it's been really difficult to get samples from other patients. We have tried getting the patients to come directly to us, but there's no real advantage. They know what they have.
Right.
So it would just simply be to aid research programs. And we did have a few MS patients that agreed to come in.
Oh, wow.
In fact, they were coming in the week that we had the Santa Clara County lockdown and Stanford labs were closed.
Of course.
And so we haven't reorganized and initiated that. In the meantime, the person who developed that is a really good developer and a very good electrical engineer, has gotten a job at UC Davis, and he wants an academic career. And that's a great position to have. But he doesn't really have a lot of access to biological stuff.
Right.
Because he's in an engineering department. Now, there still needs to be a lot done. So the nanoneedle itself is fairly inexpensive. It could be probably made for $1 to $10 wow. in fabrication. But it needs an electronic interface. We have only one electronic interface at the moment that's commercially available, and it costs $30,000. And it can only do 2 samples.
Wow.
So it's a research instrument. It's not any kind of laboratory production instrument of any kind. But it has tremendous versatility, which is why it's a research instrument. We don't need a lot of that versatility. We know what voltage to run it. We know what frequency to run it at, and so forth. So a simpler device could be built that would probably cost $100 to $200. Most labs could afford that.
Right.
Right? And even clinical labs could afford that. So he is working on a design of that in his electrical engineering department. That's a perfect thing for him to do.
Yes.
But he needs support. I have found support for him from donors who've donated money to keep him working. But unfortunately, his department has told him that it doesn't matter how much money you get donated because it doesn't carry indirect cost recovery, which the university likes to use. You will not get tenure if you don't get an NIH grant. So he's basically put on hold all of his work, and he's been writing grant after grant to try to get an NIH grant. The last one I saw from him, which was a while ago, they liked his ideas, They liked the idea of the device, but they really told him that he should not try to develop this device until he understands the biology. Well, one, there are a lot of diagnostic tests that we don't understand the biology that have been used for, decades. Especially a lot of the psychological
absolutely
diagnosis, right?
Yeah, I was just going to say there's no real biomarker for that.
So I thought that was just incredibly naive and maybe a little bit arrogant.
Yes.
It may be that they don't like MACFS research, period, because they think it's an artifact. I don't know. But the rejection held. And so he has to try to solve that. He can't solve it by figuring out the biology. He's in an engineering department.
Right.
And this is really a stupid recommendation.
Right.
That he can build this device. He's a really good designer and a really good electrical engineer. He can then give this to other labs who then can do the biology. That would be a perfect.
That would be a perfect fit then.
Yeah, that'd be a perfect fit.
Right.
And so anyway, that's where it's all held up because of the NIH.
Right.
People say, what's happening with it? Well, talk to NIH.
It's such a problem. I think, that's maybe another question is from somebody that has been in the medical field for so long, where does the stigma, where does it originate from in the medical community that ME/CFS is just, it's constantly hindered. The funding is constantly hindered by essentially the stigma that people don't seem, that the medical community has refused to take this seriously for so long. Where do you think that comes from? Does it come from just not being able to see a biomarker? Do you think that's what lended to this stigma over time?
Yeah, maybe, but also the fact that, in fact, you've seen some TV shows like this that have looked at this disease. The disease doesn't have just a simple set of symptoms. And an organ that gets affected. If it's a liver or the kidneys or the heart, it's systemic.
Right.
Right. So that's a little bit suspect. Secondly, that patients don't have, it's not a consistent symptoms. Some have this problem, some have that problem. So this variability is a real problem. I think it's all real. And I suspect it's probably genetics. That is, they have some what's called heterozygous mutations where one gene's defective, the other one is not. Well, two genes. And that makes them respond slightly differently.
Right.
And it's a systemic disease, meaning also it affects all the organs in some way or other. So it's got to be something very fundamental to the biology of humans. Anyway, that's one of the problems. And then the other problem is that since doctors dismiss it, and that's because doctors who do see the patients run their standard tests. Their standard tests are, liver function, kidney function, heart function, a few metabolites and so forth. They're all normal.
Right.
So my son, who's a severe bedbound patient, you can run those tests on him. And if you just look at the test result, and some doctors do look at it, I show it to them, they say, wow, this guy is really healthy. Right. Unbelievable. He has a heart rate of 45. He has a blood pressure of 106. Has no cholesterol. Wow. He must be an athlete.
Oh my goodness.
No, he's bedbound.
Yeah, for years.
He's not an athlete. So that's the problem with the doctors relying on those to tell them everything. And they're a very small sample set of what's going on in the body. And that's got to change. And it will change. But that's the problem, that it has the symptom variability and then no definable assays that the doctors normally do. And so because of long COVID, that's getting to be a little bit easier, and many more doctors are recognizing that this type of a disease actually exists. So it's getting better. But that's still where there's a lot of bias. There's still doctors. I find doctors at Stanford that claim, no, it's a psychosomatic disease. I know that. Partly they taught to be in medical school a little arrogant.
Yes,
And that arrogance is probably a little important for them to be able to function.
that's
Because you're
interesting.
dealing with situations that you can't control effectively, and the patients die.
Right.
And if you cared a lot about them, it would be so difficult for you to keep functioning. So they have to have this kind of protection, I think, a little bit to help them deal with human disease.
Right. The compassion fatigue, as they say with
Yeah.
doctors often.
So give them a little bit of a break, but stupidity is not acceptable in a physician.
And that is something that I hope to see change, because I think, unfortunately, just a huge barrier that continues, that people with ME/CFS still to this day will go to their doctor and will get told, oh, this is all in your head, or you're just not passionate enough. And that's for moderate patients that can make it to the doctor, right? Because people like Whitney, for example, they've been bedbound for years.
Well, and I'm lucky because I'm a scientist at Stanford. I have a good scientific reputation, and that has allowed me to really request favors from physicians, good physicians, and they often give me a break and help. And I've gotten a lot of help from really compassionate doctors, and I really have to thank them. They've made Whitney's life really at least bearable for him.
Yes, that's just so important. And I'm hoping, I think with increased medical education initiative, we could get more doctors over to that side of the
Yes.
spectrum where, they have not only compassion, but just higher level of understanding. So that if a patient comes through their doors and says, I present with these symptoms, or especially hallmark being post-exertion malaise, they're able to pick up on that sooner.
Well, and doctors have got to go back to some of the old practices of house calls.
Yes.
Stanford does allow that now. In fact, they encourage it, and they have a code for that.
Oh, do they?
It's not a matter that they're doing this kind of undercover.
Right.
They're actually encouraged to do that if the patient can't come to them. And especially with ME/CFS, they can't go to the ER.
Right.
Right. So there are patients that really cannot go there. And
Absolutely.
for those patients, house calls are called for. So anyway, but going back to the topics of, we have another observation that red cell deformability is altered in the patients. That's been a complicated one initially because there's quite a bit of variability. When we do it in the lab, it's pretty clear. But then when we try to do it with other people, like a good engineer at Stanford, and we supply the blood to them, it then comes back different. And that's been finally resolved by a professor in chemical engineering at UC Davis, Dr. Wan. And he discovered that the deformability is dependent upon oxygen concentration. So that can, and if you do it right away, you get the oxygen that's in the blood from the patient. But if you take the blood and take it over to the engineering department and then they put it in the refrigerator, who knows what the oxygen is?
Oh, okay.
And so that can account for and if it's variable, then you can't do it as a diagnostic.
Right.
So we kind of put it on a shelf. Well, that's not going to work. It's too variable. Until we started talking to Dr. Wan at UC Davis, who is pretty close. So one of our people in the lab, name is Mosen, he would take the blood and drive it to UC Davis.
Wow.
So they could get it quickly and start working with it. And they are designing a much simpler system. They initially did optics of this, and it's a good way to do it, but it's very expensive. And they're trying to do it with a very simple electrical impedance, which is what we use in our nanometer is electrical impedance.
Right.
Electrical impedance is a very good electronic way to measure things because it's very sensitive, and it's very sensitive to lots of different kinds of changes. So it's not developed yet, but the Open Medicine Foundation is actually supporting him now to develop this device. And he has graduate students and other people that are helping to work on it. We will eventually get a way to get him the blood. It looks like we can pack it in ice and get it shipped to him, and that still works okay. We'll have to make sure of that. But so he doesn't have a lot of capacity to do this. Might be able to set something up for him when the pandemic is really much clearer, where he can do it the same way we do it. A mobile phlebotomist in the area goes, collects blood, and brings it to him, and he would easily get the equipment to do the initial processing. Anyway, that's another device that is probably, he thinks he can make the device for about $1.
Wow, so it's very cost efficient.
And so, and it might be very simple. All you had to do is really plug it into a computer or something like that. But anyway, that's something that's also in the works. We have another device that I hope Open Medicine Foundation will support. It's a proposal that is into them now, which has gotten hung up with all the problems with the pandemic and so forth. But it's looking at neutrophils. And we can see a different migration rate of neutrophils when stimulated. So it's like NK cells, but it looks like it might be a much simpler assay to measure. NK cells are fine, and you can get them commercially tested, but it's a complicated assay, and that's usually messed up.
So for NK cells, so neutrophils, what exactly, just for people that may not be familiar, what exactly are they in their role?
Well, they're a type of immune cell. They're designed generally to get rid of infections. They recognize infections and can attack the cells. Young kids have a very large number of neutrophils, NK cells, excuse me. And that's because they're going to get a lot of viral infections, and they need to be able to eliminate them.
Right.
And so it is a good assay. And ME/CFS patients are less active. Now that's very similar to the neutrophils, but I'm hoping that the neutrophil assay could be made a lot simpler. And we're using probably electrolimpedance for that assay as well. It's too early to know how it's going to get designed. We don't know much about neutrophils, and that's because they're short-lived and they are destroyed by freezing. So most researchers, including here at Stanford, take a blood sample, and the first thing that they do is they'll do a limited amount of processing on it so that it will be stable, and then they freeze it. And then they take it out of the freezer when they want to do an experiment, and they put it in aliquots, and they can do, so they have access to materials all the time. They often never look to see whether whatever it is they want to assay actually survives the freezing. We know neutrophils don't survive. We know the nanonatal assay doesn't survive.
Okay, so they're very difficult to capture then.
Yes, but if you build a device that you can operate from, say, a finger stick or something of that type, and maybe one that requires minimal processing as a diagnostic is what we're thinking of. So this is not a big study of neutrophils. This is just trying to see, are there properties of neutrophils that we can use to make a diagnostic, and maybe we'll discover something about the neutrophils that have an impact on the disease itself. They're not studied very much.
Right. That sounds such an interesting and promising aspect of the disease, just because I know so many times the immune system is so implicated with ME/CFS.
Yeah. So those are all the different kinds of devices. We're very good at making very simple little devices. I'm also working with a past student of mine who's a professor now at UCLA. And I got him to start working on sweat because sweat is another material you can get from a patient. There's saliva and urine and feces and blood, but sweat is another one that you can get. Technical problem is you can't get very much of it. Well, devise very sensitive assays. The other problem is that you can't get people to be active
Right.
to sweat in order to get the sweat. Well, there's ways to induce the sweat.
Mm-hmm.
And you can do it locally. You don't have to do it systemically. So he's developed the induction methods of inducing sweat so you can get enough sweat. And he's developed, these are little devices that can go on your wrist or something of that type.
Right.
And he's trying to develop new ways to assay for molecules that are present there.
Oh,
so it could be one of the ways we would love to use it eventually. He's got more research to do, is to tell patients that they are going to crash if they continue doing what they're doing.
oh, so it's a warning system then.
Could be a warning system. That could also come from other things like heart rate, could be a warning system.
Huh.
So that's one of, and then Mike Snyder is working on those kinds of things
Right.
here at Stanford. So we would like to develop some simple thing that they could wear that tells them that if they don't stop and stop right now, if you know what I mean, from a patient point of view, you're going to crash. And I don't think you want to crash. I think it's a real problem. I certainly can make you worse, but it also might keep you in the disease.
Okay. So it might progress the disease state or just keep maintaining it.
And that was a theory that my graduate student had, who came down with ME/CFS.
Oh, wow.
She became bedbound, and then she got this theory that it was the crashing, which was
Right.
easy to do when you're bedbound.
Right.
She decided that she had to prevent crashing at all costs. She said she went for about a year and never crashed.
Wow.
And then she got over it.
Really?
Now, that's all correlational, and it's not, we don't know for sure that that's the case. She's a very, very smart scientist. And she is totally cured. I mean, totally.
That is amazing to hear, actually.
It's kind of like having a broken leg and deciding you had to go walk on it and run on it.
Right.
It'll get worse.
Yes, I often use that analogy for ME/CFS because it is like having a broken leg. And the more you keep pushing on it, because I think sometimes you see people that, especially if they're mild or moderate, they try to push through certain things.
Right.
And then you see, oftentimes it ends with them going through periods of being more severe or even bedbound. So thank you so much for joining us today, Dr. Davis. This has been really enlightening. Really appreciate all the amazing work that you're doing to help advance research for ME/CFS patients around the world.
Oh, you're welcome.